you for being here and partnering with us in this opportunity to honor the research that's taking place that our foundation is supporting, and our objective is also to inspire others to provide the funding that is so critical to institutions and individuals who really need that extra boost of support. My name is Geoffrey Baum, and I'm the Executive Director of Michelson Philanthropies. And together with my colleagues Emily Generous and Michelle Morris, it is our honor to welcome you to this evening's celebration of scientific excellence. Unlike other sessions, you are welcome to record tonight's presentations, and we'll be sharing the video online after the conference. Michelson Philanthropies was founded by Dr. Gary Michelson and his wife Alia with a daunting mission to drive transformative change in science, education, animal welfare, and social equity. Through our 4 foundations, the Michelson Medical Research Foundation, the Michelson 20 Million Minds Foundation, the Michelson Found Animals Foundation, and the Michelson Center for Public Policy, we work to advance breakthroughs in each of these areas. Whether it's advancing medical science or championing educational access for underserved populations, each foundation plays a pivotal role in shaping a better future. Tonight, we are proud to honor 2 exceptional early career investigators, Claire Otero and Romain Guyon, as Michelson Laureates. These scientists embody the bold, high-impact research that Michelson Philanthropies seeks to support. But before we proceed, I want to acknowledge 2 other guests who are here tonight. Scott Beering. Scott, where are you? There he is. He is a 2022 recipient of the Michelson Philanthropies and Science Prize for Immunology, and he's with us tonight. Scott's career has flourished since we first met him when he was at UC Berkeley. He's now opening his own lab at UC San Diego, and on a personal note, he and his wife welcomed a new baby this year. We can't take credit for that, but Mazel Tov, and that's my traditional greeting when you have a new baby, Mazel Tov, Scott. I'm also pleased to recognize Jim Crow from Vanderbilt. I saw Jim's presentation earlier. Is Jim here? He was here a second ago. Whoa. Join me in recognizing Jim. His early involvement in launching what was then the Human Vaccines Project laid the foundation for our Michelson Prize Next Generation Grants program. Their contributions, alongside the work of everyone in this room, are a testament to the profound global impact that collaborative science can achieve. At Michelson Philanthropies, we are acutely aware of the critical funding gap confronting early career scientists. Our mission is to ensure that talented researchers like Claire, Romain, Scott, and others receive the financial support and global recognition they need to advance their work and make meaningful contributions to scientific discovery. In that spirit, we'll hear from tonight's laureates shortly, but first I'd like to share a video message from Dr. Gary Michelson, whose unwavering commitment to scientific discovery and early career researchers drives everything we do. Dr. Michelson.
More than ever, we need disruptive thinkers and doers who will change the trajectory of medical science for the better. I am Dr. Gary Michelson, the founder and co-chair of Michelson Medical Research Foundation. It is beyond difficult for young scientists to get funding to conduct their own research and to do their own out-of-the-box, potentially high-risk, high-return work that would disrupt the status quo. The Michelson Prizes Next Generation Grants are changing this. In collaboration with the Human Immunome Project, we support outstanding early career investigators who want to perform disruptive research and are working to advance human immunology, vaccine development, and immunotherapy discoveries across major global diseases. When we empower young, brilliant minds and invest in high-quality, cutting-edge medical research, it's with the idea that a single scientific breakthrough discovery could affect the lives of hundreds of millions of people. So be bold, go big, disrupt the status quo, accelerate your career path, and apply for the Michelson Prizes Next Generation
That
grant.
was Dr. Michelson.
Thank you.
So thank you for listening to him. And you'll see on each of your tables there's a QR code that takes you to more information. And for those of you, because of the columns, can't see, we have screens set up in the back. So when the presentations are here, you can actually follow along if your view is obstructed. But before we go into the presentations, I want to share with you a very exciting new development here that Michelson Philanthropies is spearheading. It is the creation of a new enterprise called the California Institute for Immunology and Immunotherapy, which is currently under construction in Los Angeles. With support from the state of California and UCLA, CIII, as we call it, is set to become the global leader in harnessing the immune system to fight cancer, autoimmune disorders, and infectious diseases. This 360,000 square foot institute will integrate next generation artificial intelligence to accelerate research outcomes and is located alongside a 150,000 square foot quantum computing research center. That's the Michelsons in the building that we're actually building. We took over an old shopping mall, a 1 million square foot shopping mall in West Los Angeles that Google had begun to develop. And with the state funding, we were able to acquire it, and we're in the middle of transforming it into a research park. This combination of cutting-edge technologies, AI, quantum computing, immunology, and immunotherapy will drive faster discoveries and treatments. One of CIII's first centers is funded by a $50 million commitment from Dr. Michelson. It will focus on rapid vaccine development, a priority made ever more urgent by recent global health crises. CIII will merge AI, quantum computing, and world-class immunology to play a transformational role in the development of next-generation vaccines and immunotherapies. Now, here's the challenge we're facing right now. We're actively seeking visionary, ambitious leadership for this institute, and I encourage any of you with recommendations to share them. We have this incredible board. We just had a meeting last Thursday, and we're seeking people who want to come in and take a challenge unlike any others and transform this institution into a global enterprise. The right leader will help shape the future of immunology to revolutionize health outcomes. So now let's turn to the heart of today's program, the presentations from our 2024 Michelson Prize laureates. First, we will hear from Claire Otero, whose work with the Permer Lab at Weill Cornell Medicine has promising implications for treating diseases such as CMV, as you could read by the brochure that we have at our table. But before Claire takes the stage, let's watch a brief video introduction about her research.
My name is Claire Otero, and I'm a postdoctoral associate in the Department of Pediatrics at Weill Cornell Medicine. I received a 2023 Michelson Prize for my work in vaccine development for prevention of congenital cytomegalovirus, or CMV. I was interested in studying virology when I started graduate school, and my Ph.D. advisor, Dr. Sally Permar, introduced me to CMV immunology. Over the course of my PhD studies, I have developed a passion for CMV research, primarily because congenital CMV is so devastating for some families. But as an infectious disease, it should be preventable. Vaccine development work has focused heavily on developing antibodies that can neutralize CMV, but recent studies have suggested that other types of antibody functions are more important for preventing vertical CMV transmission. These functions involve interactions with immune cells to signal effector functions like killing of infected cells through cytotoxic granules or phagocytosis. However, CMV encodes multiple proteins called viral Fc gamma receptors that help it to evade these exact antibody functions. My research project focuses on developing vaccines that target these immune evasion proteins with the goal of improving these critical antibody effector functions. This funding enables new aspects of my preclinical investigation of CMV immune evasion proteins as potential vaccine antigens, including the development of monoclonal antibodies that will be useful as research tools and potentially even as therapeutic agents themselves. I hope that this Michelson Prize might also bring a little bit of awareness about congenital CMV and its impact. awareness of congenital CMV is low relative to its impact, and it's important for pregnant people or people trying to get pregnant to be aware of CMV as a risk to their infant so they can take steps to protect themselves.
Colleagues, please join me in welcoming the Michelson Prize laureate, Claire Otero.
Hi, everyone. Thanks, Jeff, for that introduction and making me watch that video. Super awkward. And big thanks to the Michelson Foundation for bringing me out here to give this presentation to you all. And I hope you don't mind that it's about a DNA virus. So, as was mentioned in the video, is a great introduction to this talk. I'm going to be talking about some research investigating a novel vaccine approach to prevent congenital CMV. So, CMV is the most common congenital infection, with about 1 in 200 babies born with congenital CMV in the US, although incidence does tend to be higher in developing countries. But placental transmission occurs in about 30 to 40% of seronegative women who undergo a primary infection during pregnancy, and this drops drastically to 1 to 2% in seropositive women with a secondary infection or viral reactivation during pregnancy, suggesting to us that vaccination is a promising strategy for prevention of congenital CMV because the adaptive immune response does seem to be making a difference here. But despite decades of research in this field, we still don't have a licensed CMV vaccine. And many vaccines trialed to date have focused on eliciting antibodies that can neutralize CMV. But there's this growing body of evidence to suggest that neutralization alone is not sufficient to prevent acquisition, viremia, or vertical transmission. And so we are turning towards other types of immune responses as potential immune correlates of protection. And some recent studies from a former graduate student in our group has found that FC-mediated effector functions, ADCP and ADCC, are implicated in protection from vertical CMV transmission. So turning more towards the non-neutralizing and FC-mediated effector functions of antibodies. But CMV is interesting as it's a large DNA virus. It has many, many proteins that are involved in immune evasion, and it actually encodes 3 proteins that serve to evade these exact immune functions that we call viral Fc gamma receptors, as they bind to the Fc portion of IgG, as pictured in the diagram on the right. And basically, these proteins can help the virus evade detection by the immune system through host Fc receptors through a couple of different mechanisms, including bipolar bridging, where the protein simply binds at the Fc region, and in some cases this is enough to prevent interaction by host Fc receptors on immune effector cells in the environment. But others of these proteins can actually mediate internalization and degradation of the entire immune complex, just further hiding it from the immune system. So we're interested in essentially targeting these proteins. So I'll backtrack a little bit. We know that ADCP and ADCC, as sort of examples of Fc-mediated effector functions, are associated with protection from vertical CMV transmission. And CMV has these proteins that help the virus to evade these functions. So our idea is essentially, can we improve Fc-mediated immunity by targeting these proteins through vaccination? And another key point to make here is that exceeding infection-elicited immunity has been the goal of CMV vaccine development for a long time because, seropositive women still do sometimes transmit to their babies, and so we want something that is better than natural immunity. And I'm really excited about this idea of targeting an immune evasion mechanism because I think that is a good way that we might achieve that goal. So this was essentially a proof of concept study where we started in a rabbit immunogenicity model. Because CMV is very highly species specific, we can't do challenge studies just yet, but we're looking at sort of proof of concept in this space of eliciting these FC-mediated effector responses. So we started with a protein subunit vaccine platform that's adjuvanted with a squalene emulsion Adavax. We're essentially modeling the GB MF59 vaccine, which is the most efficacious CMV vaccine that has been trialed to date. So we immunized our rabbits with 20 micrograms of GB with and without individual viral FC receptors at 20 microgram or 40 microgram doses. And we gave these in a 3-dose series each 4 weeks apart with blood draws every 2 weeks. And so what we end up with are these 7 groups where we have the GB only is sort of our baseline, and then each individual viral FC receptor at the 2 doses that we tested. And so what we find when we start looking at the immunogenicity is that the viral FCRs demonstrate immunogenicity based on just antibody binding better at the 40 microgram dose compared to the 20 microgram dose. And this does seem to be above the baseline that we see with the black lines of our GB only. And sort of, again, as we expect, the responses against GB are very similar between the vaccine groups, which makes sense given that they all received the same amount of GB. And this is the case for both GB binding and avidity, focused at the peak immunogenicity at week 10. So turning then to our FC-mediated effector functions, inclusion of our viral FC receptors does appear to elicit improved ADCP function. So this is the phagocytosis response against our vaccine-matched virus. And what we can see is that we see some improvement from the 20 microgram dose groups, particularly for GP34, which is sort of the more potent viral FC receptor. But the other 2 also elicit similarly improved responses when we get to the 40 microgram dose, which is interesting and encouraging. So, but something that's interesting when we focus on these proteins is that different human CMV isolates demonstrate a variation among these gene sequences, with GP95 encoded by the RL12 gene demonstrating extreme diversity. And among the 4 lab strains that I'm showing here, it looks like there's probably 2 isoforms that are represented among just these 4 viruses. So, but when we look at ADCP function across a few of these different strains, we actually see remarkably consistent improvement in ADCP function with each of these different virus strains for all of our vaccine groups, which is exciting to see that we can sort of confer breadth of the improvement in ADCP. So then looking at the other function of interest, ADCC, it's a little bit more difficult to interpret these results because we don't have detectable responses in our control group, the GB only. But we do see more detectable responses in our VFCR, including vaccine groups, which is an exciting start. But we hope to further this as we continue developing these vaccines. So just to conclude with our initial proof of concept study, we do see that inclusion of these individual viral FC receptors in our initial GB protein subunit vaccine does improve FC-mediated immune functions. And we note that the GB-specific IgG responses are very similar between vaccine groups, just suggesting that it is something to do with the response against the viral FCRs and not something that is changing in the response against GB. And so what we've shown so far is kind of this downstream effect of an improved FC-mediated effector response, but of course, we have a lot more work to do on this. So first, I am looking at validating the viral FCR binding responses using a Fab-only approach because, of course, the function of these proteins is to bind to FC. So we do have quite a bit of background in our ELISA that we use to test the binding. And then getting a little bit more into the mechanism of why we get this improvement. We, of course, hypothesize that there's blocking of that immune evasion, but the antibodies against the viral FC receptors could also mediate those responses themselves, and we want to get an idea of how much contribution from each of those mechanisms is happening. And then, excitingly, we have a good amount of funding, including from the Michelson Foundation, to optimize the vaccine formulation with a combination of antigens to maximize the FC-mediated response. And then once we get further down the line, we are looking to evaluate the efficacy in prevention of vertical transmission in a rhesus macaque model that is actually usable in terms of a challenge model. And then another exciting future direction that I actually don't have on this slide is the development of monoclonal antibodies that is also funded by the Michelson Foundation that we can use to study the epitopes that are important to target on these proteins and further adapt the vaccine strategies to expose those epitopes. And then additionally, those antibodies may have therapeutic use down the line themselves, which is another exciting dual purpose. So with that, I will acknowledge the wonderful mentorship and team that I have in the Permar lab, as well as our collaborators at the University of Freiburg. Philip Kolb has been amazingly supportive. He did a lot of seminal work with the viral FCRs and their basic biology. And then, of course, our funding sources, which includes a big chunk from the Michelson Foundation.
Thank you very much, Dr. Otero. Next, we're going to hear from Romain Guyon, whose groundbreaking work at Oxford is making significant enhancements in vaccine development and delivery. Notably, and I want to pass this along to everybody here and everybody that you're mentoring, Romain received his Michelson Prize before completing his Ph.D. A reminder that our grants aim to identify and support talent early in their careers. First, let's watch a brief video about Romain.
My name is Romain Guyon. I'm a PhD student in the Jenner Institute in Oxford University, and I received the 2022 Michelson Prizes for demonstration of a new single-dose vaccine technology towards a first-in-man clinical trial. I was drawn to the field of vaccine development because it is such an amazing field that involves multiple scientific disciplines and can have a major impact on global health, both at a patient level but also at the scale of a population. My research focuses on the development of single-dose vaccines, and for that, I've developed a microfluidic-based technology to encapsulate vaccine booster dose inside microparticles that will deliver this booster dose after a certain time inside the body. This approach removes the need for multiple injections, which facilitates the vaccination process for the patient. The impact of my research is to improve vaccination coverage, in particular in countries where the access of medical facilities is limited. The aim is to provide a single-dose vaccine that would ensure complete immunization to protect people's lives. The Michelson Prize is a wonderful opportunity for me to establish myself as an independent researcher. It will give more visibility to the single-dose vaccine project and open more doors for collaboration. On the project, it will allow me to assess the compatibility of the technology with different vaccines and investigate new manufacturing strategies, taking it closer to clinic.
Please join me in recognizing Michelson laureate Romain Guyon.
Thanks, Geoff, for the introduction, and hello, everyone. So I'm going to talk to you about my research, which is about developing a single-dose vaccine technology. So the concept of the work is to go from a classic vaccine regimen, which is your prime boost regimen, where you get multiple injections, to a one-shot regimen that would rely on some form of delayed delivery system that you can co-inject with the prime in one single injection. And this delivery system would remain at the site of injection, degrade over time, and release passively the booster after a certain time. So you won't need any more injection. So obviously, this would improve vaccination compliance, reduce the cost of vaccination campaigns, ease the logistics, and improve potentially the immunogenicity. So to do that, what we've developed in Oxford is this liquid core solid shell microcapsules, which is made of an aqueous core containing the vaccine, the excipients, the adjuvants. And this is surrounded by a solid shell of polymer, of PLGA, that is biodegradable and that will control the delay of delivery of your vaccine. This microcapsule is injectable, so it's below 100 microns in diameter, and it's produced by a chip-based microfluidic system, which allows us to produce homogeneously the microcapsules. So in terms of production, so this is what I've developed during my PhD. It's based on your classic double emulsification in microfluidics chips, where you inject liquid inside the microfluidic chip and forms emulsions. So as you can see in the video, it shows the process of production but slowed down, where on the left you see the formation of vaccine cores, which are then surrounded and encapsulated into a bigger droplet of solvent and polymer. And this, in real time, look like this. It's about 150 double emulsions per second in terms of production rate. And we can collect these double emulsions, extract the solvent, and it will solidify the shell and form your liquid core solid shell microcapsules. So we've put some fluorescent dye inside just to visualize the core. So as you can see, they're very homogeneous. So great, this is the method of production. Now the question is, how do they work? And to test that, we've put batches of microcapsules in an in vitro setup at 37 degrees in PBS, just to mimic the body conditions. And here you can see an SEM image of a microcapsule at day 0 of incubation. So it's smooth, it's intact, there's no signs of degradation. You can see the section where you really see the core shell architecture. But after 30 days of degradation in vitro, you really see that the polymer went through hydrolysis and it formed pores inside the shell. Again, if we look at the section, and we're just going to zoom, you really see that all these pores are now interconnected and they form a path between the inside of the capsule and the outside, which triggers the release from the microcapsule. So in terms of cumulative release, there is like 2 clear phases from this batch of microcapsules, again in vitro. First, you have a lag period where nothing is released. The polymer starts to degrade, but the pores are not interconnected yet. And then you reach a moment where the path is formed and most of the microcapsules release their content and it mimics your booster vaccination. We managed to extend this lag period in vitro again by changing the polymer that we were using as the shell material. So just by increasing the molecular weight, which increased the length of your polymer, or by changing the lactic acid content, you can move the delay from 1 to 2 weeks to up to 5 months in vitro. So great, it works well, it mimics the booster vaccination, but the big question is, does it work in vivo? And for that, we've tested the medium delay formulation, so something that was delaying the release for about 2 to 3 weeks in vitro. And we combined it with the malaria R21 vaccine that has been developed in the Jenner Institute in Oxford, and with the SMNP adjuvant that was provided by the Irvin Lam from the MIT. And for the experimental setup, we had 3 groups of interests of mice. So first, your classic prime boost regimen, where a prime dose was given at week 0, and then a second booster dose was injected at 2 weeks. We had our control group, which was the prime only. We received both the prime and the booster dose at week 0 in one single injection. And then finally, our group of interests, which was the prime and microcapsules, with both the prime dose and the booster dose, but encapsulated inside capsules, that was injected at week 0. All of this was dose matched. We monitored the immunogenicity, and we also challenged the mice with malaria to assess the efficacy of all regimens. So in terms of immunogenicity, so here you can see the different antibody titers evolution over 11 weeks for the 3 regimens, and then a comparison at week 6. For the first 2 weeks, they behave the same, and it makes sense. It's due to the effect of the prime dose. But then the prime-only regimen titers plateau, while the 2 other regimen continue to rise. And this is due to the effect of your booster injection for the booster group and due to the release from the microcapsules for the microcapsule groups, which shows that the microcapsules are able to mimic this booster effect. And in fact, it managed to increase the antibody titers by about 20-fold compared to the prime-only. In terms of efficacy, we challenged the mice, we exposed them to malaria at different time points for these 3 regimens, and we looked at the proportion of survival for each groups. So this is represented by the bars that you can see here. And we use all this data and combine them into a log binomial regression model just to compute the evolution of efficacy over time. As expected, the prime-only regimen decreased with time because the prime effect just fades with time, while our prime boost's efficacy remained stable over time at around 70%. And for our group of interests, we had a higher efficacy than the prime-only, and it continued to rise consistently to almost reach the level of protection given by the prime boost. So this was for the proof of concept with malaria. And now for my last slide, I'm just going to show you a small overview of the different aspects that we are now investigating, so which are different key features that would be important for the deployment of the technology. So first, injectability. We made sure that these microcapsules could be injected through a 25G and even a 27G needle, which is what's allowed for humans intramuscularly or SC, sub-Q. We also are now investigating the potential to extend the shelf life of these microcapsules by drying them. So that's the image that you can see in the middle of the slide. And they, due to osmotic effect, the capsules lose temporarily their shape, but when you rehydrate them, they just recover it. We're also trying to investigate other vaccines, so trying this technology with the rabies vaccines for post-exposure prophylaxis. We're playing with different excipients, sugar, hydrogels to try to assess the stability of what we put inside. And finally, we're trying to scale up the technology, to scale up the production by playing either with the formulation of the flows that we use in the microfluidic process. So these are the 3 videos that you can see where we just decrease the interfacial tension of some flows to increase the generation rate of the microcapsules. And we also are now trying to parallelize the system so we can just increase the throughput. So I'd like to thank everyone that's involved in this project and obviously the founders, so the Michelson Foundation and the VaxUp Global Consortium. Thanks very much for your attention.
Just curious, from a global health perspective, what species of malaria did you test against so far? And I'm assuming based on the shelf storage figure that you had, the dried capsule, they can be stored at room temperature?
Yeah, so it's against falciparum. That's the model that we have. And in terms of storage, so we actually tested the storage in liquid. So the storage is first to preserve the ability of the microcapsules to degrade over time and release, because the polymer, the PLGA, degrades when it's in water. So if you store it liquid, it starts to degrade. But what we've tested is keeping it at 4 degrees in liquid. It was already fine. For other vaccines, that would be a limitation just because of the storage of the vaccine that is encapsulated inside the capsules. And that's why we tried to dry them. But yeah, what we've tested so far was at room temperature.
Super exciting stuff. Just curious, have you tried empty microcapsules along with just to try to control for, like,
yeah,
is there an adjuvant effect of the microcapsule itself?
So no, so yes, we tried, and no, there is no adjuvant effect. Sorry. We tried with the microcapsule. They are too big, so they're not uptaken. That was the point of making them big enough so that they are not uptaken by any cells. So they don't really contribute. They just release. So by their kinetics, they will have some form of potential adjuvant effect, and that's why we're trying to see if they could actually be even better than a booster, which is right now not the case with malaria. It seems to match pretty well in immunogenicity, but not getting better. But because they're too big, yeah, they're not like uptaken, so they don't seem to contribute when you co-inject them with a prime. Thanks.
Our thanks to now Dr. Guyon. In closing, I want to extend my sincere thanks to Debbie and Amanda and the entire Keystone Symposia team for organizing this exceptional gathering, and to our conference chairs, Kazmekia Corbett, William Klimstra, and Yolanda Schmidt, for assembling such a remarkable program this week. The work of the researchers here, everyone in this room, is crucial to advancing the frontiers of science, and it's our privilege to support this vital mission. I want to thank you for joining us. I have a special announcement. We were able to convince the bar to extend for one more drink since we're having the program. And I look forward to, we all look forward, on behalf of Alia and Gary Michelson, to continuing this collaboration for the betterment of human health. Thank you and have a wonderful evening.